Good afternoon, everyone. First of all, thank you for giving me an opportunity to present one of our study. My talk is about the enhancer. Enhancer, as we all know, is a type of the regulatory elements and who play the very important role to define the specificity, cell or tissue specificity in different development stage. This study, we have the particular interest in a group of the enhancer we call it tele-enhancer. So the tele-enhancer in our study, we mean the enhancer. Enhancer 2, like the enhancer 2, which are separated from the target gene by the intermediate gene which are not expressed in the particular scenario. And by contrast, the proximal enhancer means the enhancer which located in the neighborhood of the target gene. So we start off our study with the human heart enhancer. Okay. So the enhancer here are identified in the P300 chipset data, and we have like thousands of the heart enhancer. And the heart gene we collected based on the gene expression data and the gene autology annotation. The gene which are all expressed in heart and all annotated with the heart development, we call it heart gene. So after place the heart enhancer and the heart gene along the chromosome, along the whole DNA, whole genome, we then connect the enhancer, each of the enhancer to two nearest heart gene, one located upstream of enhancer, the other one located downstream enhancer. So based on this map, we can categorize the heart enhancer into two groups. One is tele-enhancer. The other one is proximal enhancer, as we defined in last slides. So before we move to investigate the tele-enhancer, we first validate the connection, the gene and the heart connection we just built. So we turn to the information from the third party. First of all, we look into the synthetic blocks. We go into the synthetic block. Synthetic blocks are built based on the alignment between the human genome with another species genome, and then we hope that the enhancer and the target gene are located in the same synthetic blocks. Which means we hope that the enhancer and the target gene between them they have the few breakpoint as few breakpoint as possible. So we try to use the synthetic blocks which built based on the human, based on the alignment between the human genome with another vertebrate genomes, which including the chimpanzee, macaque, mouse, cow, and chicken. And so for all of the scenario we used, we found out the breakpoint between the enhancer and the target gene are significantly low than expected. So either in the tele-enhancer case and also in the proximal enhancer case. Also, we look at the DNase regulator blocks, which built based on the DNase chipset data, which are available in the INCOM project. And then we found out the the heart enhancer and their target gene are more likely located into the same regulatory blocks. So no matter it is the tele-enhancer or the proximal enhancer. So all of the evidence supports that the connection we built between the enhancer and the gene makes sense. So we move to the next step to look at the enhancer. Immediately we found out the more than half of the heart enhancer are tele-enhancer. This is a little bit surprise to us. Is a large portion, and likewise we found out the heart gene. More than half of the heart gene are regulated completely regulated by the tele-enhancer, which we call such kind of gene as the gene T, and the gene which have at least one proximal enhancer, we call it gene P. So these two pie charts show that the tele-enhancer plays a very important role in the heart development. So it's worth to probe the tele-enhancer in depth. We first look at the function of the tele-enhancer, and then we find out both tele-enhancer and the proximal enhancer, both of them have the close relationship to the heart development, which are not surprised because we working on the human fetal heart. But on the other hand, we find out only the tele-enhancer plays a unique role. It's involved in the biological process like the oxidative phosphorylation and the mitochondrial organization and the energetic biological process. So we make the speculation that because all of this biological process is somehow is very basic biological process. So we make the speculation here is that maybe the tele-enhancer more associated with the low tissue-specific function, and the proximal enhancer have the more high functional specificity. So we're looking at the relative gene expression, the relative gene expression of the gene T and the gene P, and found out that the gene P have the very high, have significant high relative expression, which show that the gene T or the tele-enhancer plays a low tissue specific function. We're also looking at here we also found out some of the biological keywords, the keywords mitochondrial. So we collected all the genes which are related to the mitochondrial bio process, and found out the tele-enhancer are highly related to the mitochondrial process, but the proximal enhancer are not. So all of this process indicate, all of this evaluation indicate that tele-enhancer are related to some basic biological process which have the low tissue specificity. And our finding about between the tele-enhancer and the mitochondrial biological process we found out some support to our finding here. And after find out the functional difference between the tele-enhancer and the proximal enhancer, we suspect that we speculate that the different group of the enhancer may be activated by the different regulatory network. So we're checking out the TF binding motifs, which are enrichment in the different group, enhancer group. then we found out both of the enhancers are enriched for very well-known cardiac transcription binding site, like MEF2, but the tele-enhancer and the proximal enhancer have the unique binding motif pattern. For example, only the tele-enhancer are enriched for the TD1 and NKX 2.5 and TBX5, and the proximal enhancer are only enriched for the NFAT and SRF, and like GITA4 and CRB like P300. So this map show that the tele-enhancer and the proximal enhancer, on the other hand, they share some of the binding motifs, but they also show the distinctive binding motifs. We also look into the evolution of the enhancer. Before we go to the to study the evolution, we first find out the tele-enhancer and the proximal enhancer. They show the very similar GC content and very similar CpG site density. So these two fingers help us to eliminate some confounding factor which may lead to the which may have impact on the evolution of the enhancer. So we look at the difference between the evolution difference between the tele-enhancer and the proximal enhancer. And we find out, first of all, the tele-enhancer are enriched for the conserved elements. The conserved here we identified based on the FastCon score. So the FastCon score is estimated based on the 44-way vertebrate species. So it's very long evolutionary history. And then we look at we zoom our study into the primate evolutionary history, and then we find out this dashed line present the natural selection. So the area below this dashed line means the negative, and above means the positive. So we found out both of the tele-enhancer, proximal enhancer, and a very strong negative constant. But the tele-enhancer have even more stronger the selective pressure than the proximal enhancer. Also, we look into the SNP distribution, which present the selection pressure on the enhancer in the modern human history. Then we find out the tele-enhancer have the low SNP density and also have the very high preference to the low derivative allele frequency SNPs. All of these results show that the tele-enhancer have the strong impose the strong selection pressure in the different evolution history. And then we also expand our study to the other tissue. And then in this tissue, the brain and the lung tissue, the enhancer was identified based on the histone mark, the enhancer specific histone mark. And the gene are also based on the expression data and also based on the gene ontology. So it's very similar to what we found in the heart. The half of them, the enhancer are tele-enhancer, and half of the gene are gene T, regulated based on the tele-enhancer. And also the function, we found out both of the tele-enhancer and the proximal enhancer have the tissue-specific function, and the but the tele-enhancer have the specific function in very basic biological process. And then we found out the similar to the heart, the gene T, so a little bit low, tissue specificity. And then the similar, the tele-enhancer are conserved in the term of the SNP distribution in the brain tissue and the lung tissue. And then the other, just 2 more slides. I'm sorry, 2 more slides. So we also do the extend our study to the 7-cell line, which the enhancer and the gene expression data are provided in the ENCODE project. And then we found a similar thing. The enhancer, the tele-enhancer have the low functional specificity and also are much more conserved than the proximal counterpart. So yeah, the same thing, the SNPs, the SNP distribution, we get very similar, we found out a very similar trend, the similar trend to the heart tele-enhancer. So the conclusion here, based on the genome-wide map of the tele-enhancer in the different, we established, we found out the tele-enhancer in the different tissue. And then we do the very systematic and comprehensive study on the tele-enhancer, find out it's conserved than proximal enhancer and have the low tissue specificity and have the distinguished recruit the distinguished transcription factor from the proximal counterpart. And thank you for your attention.